Fetal membranes are also known as extraembryonic membranes, are associated with the developing embryo or fetus and protects and nourishes it during gestation. There are mainly four extraembryonic membranes: the amnion, the yolk sac, the chorion, and allantois. Placenta is the most important part of development during the fetal period. It is also included in the fetal membranes, which is derived from the extraembryonic tissues. Connecting stalk or umbilical cord, which mainly connects the placenta with the fetus. The important functions of fetal membranes are protection, nutrition, respiration, excretion, and synthesis of hormones. Placenta and fetal membranes. The basic structures which are derived are amnion, which is mainly derived from the epiblastic layer, and extraembryonic mesoderm. Yolk sac is derived from hypoblastic cells and also extraembryonic mesoderm. Allantois, a diverticulum from the hindgut, is derived from the ventral wall of hindgut, or in other words, endodermal in derivative. Chorion, derived from the trophoblastic cells and extraembryonic mesoderm. The trophoblast which forms the chorion is cytotrophoblast and syncytial trophoblast. Lastly, coming to the most important part, that is the placenta, which has fetal portion derived from chorionic frondosome and maternal portion which derived from the maternal decidua, that is the decidua basalis. Coming to the first part, that is the umbilical cord, the development of umbilical cord. Umbilical cord is 1 to 2 centimeters in diameter, 30 to 90 centimeters in length. It has a smooth surface because it is covered by the amnion. Umbilical cord is enveloped by amniotic membrane. At full term, one end of umbilical cord is attached to the center of anterior abdominal wall of the fetus. The other end is fixed to the fetal surface of the placenta, usually central in position. Attachment of umbilical cord normally is attached to the point nearer to the center of the fetal surface of the placenta. Therefore, umbilical cord is an important lifeline that connects the fetus with the mother. Umbilical cord is derived from the connecting stalk, and connecting stalk is formed at the end of second week of development. When blood vessels appear in the connecting stalk, that is around third week, connecting stalk forms the umbilical cord. Umbilical cord finally connects the fetus with the placenta. As the germ disc bends ventrally during the formation of embryonic folds, that is the head fold, tail fold, and right and left lateral folds which occur during the third week of intrauterine life. The amniotic cavity enlarges and surrounds the fetus. Further development of amniotic cavity enlarges rapidly at the expense of chorionic cavity. Amnion begins to envelop the connecting stalk, the yolk sac, allantois, all of them crowding together at the ventral part of the embryo. Embryonic folds, head and tail folds and right and left lateral folds converge on the ventral surface of the fetus, forming a primitive umbilical ring which occurs around the fifth week of intrauterine life. So around fifth week of intrauterine life, structures passing through the primitive umbilical ring, that is passing through the umbilical cord. It is the connecting stop, that is when it is invaded by blood vessels, it forms the umbilical cord, two umbilical arteries and one umbilical vein. Arteries carry deoxygenated blood and the vein carries oxygenated blood. Primitive umbilical ring also presents the yolk sac or vitellointestinal duct, which is connected with the definitive yolk sac. Primitive umbilical ring has the allantois, distal part of allantois. The fourth important structure which is present through the primitive umbilical ring is the intestinal loops. These intestinal loops herniate and they herniate into the umbilical region, and this is known as physiological hernia. It is due to the small abdominal space. It herniates into the umbilical region. By 10th week, these intestinal loops return back to the abdominal cavity as the abdominal cavity enlarges. The fifth important structure which is seen in the primitive umbilical ring is the extraembryonic part, which connects with the intraembryonic part of coelomic cavity. 12th week or third month, the umbilical cord presents the following structures, that is the amniotic cavity obliterates the chorionic cavity and yolk sac shrinks gradually and obliterates. Changes occur in the gastrointestinal system. Therefore, allantois, vitelline duct and vessels get obliterated. Umbilical cord, third month or 12th week of intrauterine life, therefore consists of umbilical vessels, that is umbilical vein and umbilical artery, surrounded by primitive mesenchymal cell and Wharton's jelly. No nerves are detected because umbilical veins are longer than the cord. Twisting and bending of the vessels are common, forming loops and producing knots. So, contents of umbilical cord at full term consists of two umbilical arteries which are derived from ventral division of internal iliac artery, convey deoxygenated blood from the fetus to the mother. One umbilical vein. Early in pregnancy, veins are two. Right umbilical vein disappears and the left umbilical vein persists, carries the oxygenated blood from the placenta to the fetus. Wharton's jelly. Primary mesodermal cells undergo mucoid degeneration and forms Wharton jelly, which protects the umbilical vessels, surrounds the umbilical vessels and protects the umbilical vessels. At birth, umbilical cord is ligated. The circulation of blood in the umbilical arteries is first stopped by reflex spasm of the muscular wall, followed by cessation of blood flow in the vein. Coming to the applied concept of umbilical cord, normal umbilical cord usually about 30 to 90 centimeters. Short umbilical cord may cause difficulty during delivery by pulling the placenta from its attachment in the uterus. that is premature separation of the placenta, causing intrapartum hemorrhage. Extremely long umbilical cord. Extremely long umbilical cord may encircle the neck of the fetus and cause strangulation, or may have tendency to prolapse, causing compression or hypoxia to the baby. Cord prolapse. Knots of the umbilical cord. True knots when the fetus passes through a loop of cord. If pulled tight, fetal asphyxia may result. False knots are location a collection of Wharton's jelly containing a loop of umbilical vessels. False cord knots are not very important. True knots are important where it deals with the fetal asphyxia. So false knots look like tortuous structures but do not cause any harm to the fetus. True knots, on the other hand, are very dangerous and may cause obstruction of blood flow in the umbilical vessels, leading to fetal anoxia and fetal death. Torsion of the cords. An excessive twisting of umbilical cord, it can occur at any site in the umbilical cord. It is considered to be severe form of cord constriction. Umbilical cord torsions are considered to be sporadic event and a rare cause of intrauterine fetal death. So twisting or torsion of umbilical cord along the left upper extremity causing strangulation of the umbilical cord. Hematoma. This is usually present at the fetal end of the cord, a reddish purple swelling of variable size. It's an extravasation of the blood, mainly venous, sometimes arterial, in the Wharton's jelly, identified by ultrasound, and may result in intrauterine fetal death. Abnormalities of the umbilical vessels. So umbilical arteries are two in normal and vein is one. Sometimes single umbilical artery and one umbilical vein is associated with the umbilical cord. In such cases, one umbilical vein and two umbilical arteries are normal. One umbilical vein and two umbilical arteries are normal. When there is one umbilical artery, it is considered as abnormal and is associated with cardiac, vascular defects and other anomalies. Single umbilical artery is associated with cardiovascular defects and other anomalies. Battledore placenta, wherein the umbilical cord is attached to the margin of the placenta. It's not that dangerous, but sometimes it can lead to dangerous effects. Velamentous placenta, where the umbilical cord is attached to the amnion away from the placenta, is attached to the membranes. It is dangerous for the fetus due to the rupture of blood vessels during labor and because it's not protected by Wharton's jelly. Coming to the applied concept of umbilical cord, anomalies or abnormalities of umbilical cord can be identified by color flow Doppler ultrasonography used during prenatal diagnosis to identify the position and structural abnormalities of umbilical cord and its vessels. Next, coming to the fetal membrane, that is the amnion and the amniotic cavity. Amniotic cavity surrounds the fetus and protects it from external shocks and jolts. Amniotic cavity appears during the second week of development. It's present between the germ disc and the trophoblastic cells, cytotrophoblastic cells. And it appears development of amniotic cavity starts as early as the 8th day of intrauterine life after the implantation of blastocyst, which occurs around the 7th day or end of first week of development. Coming to the important stages of development of amniotic cavity, which occurs during the second week, develops from the trophoblast, which is between the epiblastic cells and extraembryonic mesoderm. Eighth day, the epiblastic cells and the extraembryonic mesoderm are responsible to formation of the amniotic cavity, which forms around 8th to 9th day. 14th day, the amniotic cavity is surrounded by, it's roofed by amnioblastic cells, which are derived from the trophoblast, and floored by epiblastic cells. Amniotic cavity enlarges at the expense of fetal folds, embryonic folds into the chorionic cavity. Amniotic cavity, which is formed during the second week of development. During third week of development, embryonic folds are present. They start developing, that is the head fold and the tail fold. By fourth week, the amniotic cavity, due to this folds, enlarges. By fifth week, almost it enlarges into the chorionic cavity. And finally, obliterates by end of second month or eighth week. It finally obliterates the chorionic cavity and completely surrounds the fetus. Once again, the amniotic cavity enlarges at the expense of fetal folds, embryonic folds into the chorionic cavity, and later it completely obliterates the chorionic cavity and also the lumen of the uterine cavity. As it fuses with the chorion, it is known as chorioamniotic fusion. Amniotic fluid is clear and watery, produced partly by the amniotic cells and mainly derived from the maternal blood. Amniotic fluid increases from 30 ml at 10 weeks to 450 ml at 20 weeks, 800 to 1000 ml at the time of 38 weeks. Fetus is suspended by the umbilical cord in the amniotic fluid, which acts as a protective cushion to the fetus from external jolts. Factors which regulate the volume of liquor amnion. So at the beginning of fifth month, the fetus swallows its own amniotic fluid and drinks about 400 ml per day. It is absorbed by the gastrointestinal system or GAT. Then it enters the bloodstream. It then passes to the maternal blood via the placenta. Fetal urine is also added daily to the amniotic fluid from fifth month by fetal kidney. But this urine is mostly water because placenta functions as an exchange for metabolic waste. So fetal swallows amniotic fluid around 400 ml per day. At the same time, it also secretes urine and which is mostly water into the amniotic fluid. Both occur at the beginning of fifth month of intrauterine life. Coming to the circulation fate of amniotic fluid, which remains constant in balance. Most of the fluid is swallowed and few passes into the lungs of the fetus and absorbed by fetal blood and therefore finally by the maternal tissue or the placenta. Part of the fluid passes through the placental membrane to the maternal blood in the intervillous spaces. Other part of fluid is excreted by fetal kidneys into the amniotic sac. Functions of the amniotic fluid. It absorbs jolts of the fetus and prevents injuries, prevents adherence of the embryo to the amnio, allows free movements of the fetus aiding muscular development, maintains symmetrical external growth and differentiation of delicate tissue of the embryo, mainly the lung tissue, assists in maintaining hemostasis of fluid and electrolytes. Finally, it permits the amniotic fluid, permits the various important studies on enzymes and hormones and diagnosis of fetal sex and chromosomal abnormalities. Abnormalities of amniotic fluid. Polyhydramnios or high volume of amniotic fluid which exceeds more than 2 liters. It occurs when the fetus is unable to swallow in esophageal atresia or anencephalic babies, where swallowing reflex is absent, or maternal diabetes. Oligohydramnios is decrease of amount of fluid less than 400 ml, and it may cause mainly due to the renal agenesis. Oligohydramnios can cause clubfoot, lung hypoplasia, facial defects, and premature rupture of amnion. Premature rupture of amnion is a common cause of preterm labor. Causes the rupture sometimes may be due to trauma. Amniotic band syndrome. Amniotic bands or tears in amnion is mainly due to the infections and toxins in the amniotic fluid. They form rings around the limbs or fingers, causing physiological amputation of that part. Amniotic band syndrome, where various fibrous bands of amniotic sac get tangled around the developing parts of the fetus, restricting the blood flow and causing amputation. Etiology of amniotic band syndrome remains unknown, but however it may be due to the trauma. It can be detected by ultrasound. Clinical importance of amniotic fluid. Amniocentesis or aspiration of amniotic fluid through the cervix or anterior abdominal wall. This test is usually done between the weeks 15 to 20 of pregnancy, where the amniotic fluid is roughly about 500 to 400 to 500 ml, mainly done for nuclear sexing of the fetus in sex-linked diseases, estimation of enzymes in case of gross fetal abnormalities. Amniocentesis is done under the guidance of ultrasound. Coming to the next important membranes, fetal membranes, the yolk sac. The yolk sac is attached to the embryo, formed by the cells of hypoblastic layer of a bilaminar embryonic disc. Yolk sac is formed during the second week of development. Primary yolk sac is formed around the 10th day of intrauterine life. Secondary yolk sac is formed around the 14th day of intrauterine life. Yolk sac is situated in front or the ventral part of the embryo. It's lined externally by splanchnopluric extraembryonic mesoderm. Development of yolk sac, that is primary yolk sac, which starts around the 9th day of intrauterine life. At the embryonic fold, flat cells originate from the hypoblastic layer, and these cells form a thin membrane, and this is known as House's membrane, which lines the inner surface of cytotrophoblast. Hypoblastic cells, together with House's membrane, is the primary yolk sac cavity. 14th day or the second week of intrauterine life, secondary yolk sac is formed, which is smaller than primary yolk sac within the extraembryonic coelomic cavity. It's also derived from the cells from the hypoblastic layer. Yolk sac cavity is incorporated into embryo due to the embryonic folds to form the primordial gut during the third week of embryonic life. So this is the embryonic part, second week, formation of yolk sac, primary yolk sac and secondary yolk sac. Third week is the formation of embryonic folds where most of the yolk sac is taken inside by the embryo, forming the primitive gut tube. And primitive gut tube is therefore formed by the embryonic folds, the incorporation of yolk sac by the embryonic folds. So once again, to see the development of primitive gut tube, the flat trilaminar disc folds, head fold and tail fold, and also lateral folds. And with these folds, the yolk sac is incorporated into the embryo to form the primitive gut tube. The primitive gut tube is divided into three regions: foregut within the head fold, hindgut within the tail fold, and midgut communicates with the yolk sac by the vitellointestinal duct. The part of yolk sac which is not incorporated into the embryo, which remains outside, is known as the umbilical vesicle or definitive yolk sac. By 10th week, that is the definitive yolk sac or umbilical vesicle. 10th week umbilical vesicle shrunkens to pear-shaped remnant and is connected to yolk midgut by the yolk stalk. It atrophies as a pregnancy advances. It gets fibrosed and degenerated by the 20th week of intrauterine life. So therefore, fate of the yolk sac. Fate. The yolk sac forms part of the yolk sac which is incorporated in the embryo forms the gastrointestinal system or GAT, forms the foregut in the head fold, tail fold, hindgut, and the intermediate part is the midgut which is connected to the yolk sac. And part of yolk sac which remains outside the embryo finally degenerates by the end of 20th week of intrauterine life. Coming to the important roles and functions of the yolk sac. The most important function of the yolk sac is nutritive or metabolic organ during the early stages of development of the embryo prior to the development of blood vessels. It also contributes for the first blood cells which appear along the splanchnopluric mesoderm lining the yolk sac. It forms the primitive gut tube around the end of 4th week, and also from the primitive gut tube it also forms the respiratory system. Caudal part of the yolk sac, that is from the hindgut, arises the primordial germ cells which are the stem cells which finally form oogonic and spermatogonic cells. Yolk sac is also the source of extraembryonic mesoderm. It's important for embryonic patterning or patterning, establishing the blueprint of organism body structure. These are the important functions of the yolk sac. Coming to the next part of fetal membranes, that is the chorion. Chorion also develops during the second week of intrauterine life, basically derived from extraembryonic mesoderm. It surrounds the fetus and it also takes part in contributing for the nutrition of the fetus in the early part of development. Chorionic cavity is lined by extraembryonic mesoderm and it develops around the second week of intrauterine life. Chorionic sac therefore consists of the chorionic cavity therefore consists of internally it's lined by splanchnopluric extraembryonic mesoderm lining the secondary yolk sac. Externally it's lined by three layers: somatopluric layer of extraembryonic mesoderm. External to it is a cytotrophoblast and the outer layer is a syncytial trophoblast. These layers form the external lining of the chorionic cavity. These three layers together, they form the chorionic plate from which the villi arise, and therefore the villi are therefore known as chorionic villi. Chorion forms the stem villi. From the chorion arises the villi, and these villi are the stem villi or anchoring villi or the tertiary villi. Chorionic villi at the embryonic pole forms the chorionic frondosome, the fetal part, which is overlapped by the decidua basalis. Together they form the placenta. Chorionic tertiary villi therefore differentiates into chorionic frondosome overlapped by decidua basalis at the embryonic fold. Together they form the placenta. At the embryonic pole, the villi are degenerates and it is known as chorionic levee, which is overlapped by decidua capsularis. Stem villi extending from the chorionic plate to the decidual plate. Chorionic villi is highly bushy in the early part and it is the chorionic frondosome. At the embryonic, coming to the clinical correlation of the chorion. Chorionic villus biopsy is done to detect various genetic disorders of the fetus, and it's also done under the guidance of ultrasound, done between 8 to 10 weeks of intrauterine life. The fifth important fetal membranes is allantoic diverticulum. Allantoic diverticulum arises from the ventral wall of hindgut, extends from the posterior intestinal pole, the hindgut, to the cloacal membrane, and from its ventral wall arises the allantoenteric diverticulum, that is the end of third week of intrauterine life, and it enters the connecting stalk. Allantois is a reservoir for excretory products in the lower animals. Appears like a sausage-like diverticulum from the hindgut extending into the connecting stalk. Allantois invades the connecting stalk that suspends the embryo in the chorionic cavity. Connecting stalk later forms the umbilical cord. It's involved in early hematomysis. Allantoic blood vessels, artery and vein later becomes the umbilical vessels. Remnants of allantois becomes urachus, a ligament which connects the umbilicus to the bladder in adult. Allantois from the ventral wall of hindgut. Its vessels finally contribute to form the umbilical vessels, the umbilical artery and the umbilical veins. 10th week, the extraembryonic part of umbilicus degenerates and intraembryonic part of allantois connects to the apex of the bladder with the umbilicus. As bladder enlarges, allantois forms a thick tube which is the urachus. After birth, urachus forms a fibrous cord which is known as median umbilical ligament. So coming to the various stages of development of allantois. Allantois appears as a diverticulum from the caudal part of yolk sac which forms the hindgut. Its extraembryonic part degenerates and intraembryonic part is connected to the developing urinary bladder. Connects the urinary bladder to the umbilicus and that is known as urachus. After birth, the urachus is obliterated and fibrosed and becomes the medial umbilical ligament. Therefore, the functions of allantois is early stages. It forms the blood formation in its wall during 3rd to 5th week. Its blood vessels finally persist as umbilical vessels, the artery and the vein. The fate of allantois in humans. 3rd week, Allantois appears as a diverticulum from the hindgut or the caudal part of the yolk sac, which enters the connecting stalk. Around ninth week, the allantois is connected to the vesico-urethral canal, which later forms the urinary bladder and urethra. Around third month, it's, it's completely gets fibrosed and it's connected to the apex of the urinary bladder. Adult urachus becomes fibrosed and it's connected to the apex of the urinary bladder to the umbilicus. Now coming to the various important remnants, that is urachus, that is the allantois, which becomes fibrous, connects the apex of urinary bladder to the umbilicus. It lies in the median umbilical fold and it retains as median umbilical ligament. Next we have folds lateral to the medial umbilical folds. These are medial umbilical folds and these are the remnants of umbilical arteries, right and left umbilical arteries. Umbilical vein is represented as round ligament of the liver.